Cancer is commonly known as the uncontrolled division of abnormal cells in any part of the body. The mechanisms of cell abnormality vary with acquired or inherited influences common. Our environment can also facilitate cancer, from increased levels of radiation to carcinogenic exposure. Though these external factors are known to increase incorrect cell replication, they are common and natural. Oncogenes are known as cancer-causing genes in the genome and are activated via chromosomal rearrangements and gene duplications. Due to these genomic alterations, oncogene activation occurs, creating cells which duplicate at rapid rates. Though not ideal, the human body created the guardians of the genome, p53, a tumor suppressor. Tumor suppressors reduce cell division rates, repair DNA mistakes, and ensure cell death is executed. The inactivation of tumor suppressors, again through genomic alterations, limits the body's ability to remove abnormal cells. The inactivation of these tumor suppressors can only occur if a mutation on both locus on separate chromosomes occur. Here's a simple analogy. Imagine the 3 best genetic students in Australia in an empty lecture hall. Each student represents a normally functioning cell, undergoing normal replication. The most talented student encounters a somatic mutation in one of his repetitions. This mutated cell experiences extra metabolic activity, replicating at a rapid rate. Due to the tumor suppressor being inactivated, the star student is left to continuously replicate, becoming cancerous.
So how does one sequence a cancer? What is unique about the sequencing of cancer is that it uses whole genome sequencing, or WGS, not just whole exome sequencing, or WES. Unfortunately, the extra information that WGS provides, which is useful in a theoretical scope, results in seemingly endless identification of DNA variants. If we're looking for a cancer, we simply want to identify the somatic mutations. In essence, a functional somatic morphology is a veritable needle in the haystack of a completely sequenced genome. Hidden amongst innocuous variations, it becomes significantly harder to identify. But there is a way around this. Using a standard template sequence called the human reference sequence, the sequenced DNA of the individual in question can then be assembled from raw DNA sequences. This produces a clear link between the genomic coordinates and the respective DNA bases using computer-intensive programs. Then, after this, the sequence is rid of any irrelevant DNA which does not directly relate to the somatic variant. This is simply done by comparing the cancer genome of a given patient to a tumor-free sample from the same person. This sample is known as a baseline dataset and is used as a subtraction factor in order to outline all the acquired somatic mutations. Imagine you have 2 burgers. Your beef burger represents your tumor-free sample, and your vegan burger represents your cancer genome. By comparing them and identifying the germline genome, which is denoted by the buns and fillings, only the patty is left. This patty represents the somatic genome. The difference in between the beef and the vegan patty is reflective of the somatic mutations that the cancerous cells picked up.
Whole genome sequencing and exome genome sequencing are known as next-generation sequencing, as the genomic technology utilized has become vital in cancer research today. As cancer is an evolving disorder, next-generation sequencing can monitor the score tumor, progression, remission, and relapse. This is done by analyzing the genetic content at different time points. By monitoring cancer at these certain time points, provides the cancer research community with ever-growing knowledge of that cancer and this also may lead to more effective drug decisions, better patient treatment options, and more accurate prognosis. The key application of whole genome sequencing and exome genome sequencing is that it has emerged as the most ideal method for discovering somatic mutations by pairing the tumor and normal whole genome sequencing or comparing the normal and tumor exogenous sequencing. This thus assists in the discovery of what is a driver mutation and how that tumor cell behavior may alter the passenger tumors, mutations, and how this may spread throughout the body. Next generation mutation sequencing is useful in most cases, as a mutation tends to affect common genes, highlighting the dysregulation of shared important pathways for tumor progression. Hence, by doing this, we can take several cases of the same tumor and eliminate DNA changes that are not relevant, thus allowing us to find the most tumorigenic pathways. For our genetics topic, we chose to look at genome sequencing of cancer. We chose this topic because we were curious about how using genome sequencing to find the exact mutation has actually occurred and how this impacts the cells around it. As well as also interesting to see how technology has advanced and how it's impacted on diagnosis and care of individuals with cancer. As a team, we worked really well together as we separate the research into 3 areas and each investigate our own area. So our own separate area. And then at the end of the day, we all came back together and we cemented ideas together to figure out the goal and game plan of it. I find it really difficult personally researching the applications of genome sequencing and choosing what specific technique we wanted to use, just as there was a lot of information on all the techniques in general, but narrowing it down to whole genome sequencing and exome genome sequencing was a bit more tricky, trying to find just applications for it. But at the end, we did get there. My role in the group was to research genome sequencing and its applications and how it has developed. As well, we all equally contributed to creating this video.
So we came up with the idea of cancer sequencing research just because we think it affects, cancer in itself affects so many youths of Australia, and we wanted to aim a video at high school students dealing with cancer, with a family member who has it, or even just as a learning tool through high school. So the most challenging part for me in this was my job was to search what cancer is and how does it form, and there are so many different ways that can happen. So I had to really get in depth so I could surmise it and kind of make sense at the same time, which was really good. That was a bit of a challenge as well. But all in all, our group was really good. We worked really well, and we were lucky enough to enjoy our time together. So it wasn't a huge pain to kind of come in and have to deal with them, and it's quite rare at uni, so I'm pretty stoked with that. Yeah, and I learnt much more in depth about cancer and how to maneuver through iMovie and not stuff it up, which was cool too.
The way we came up with this idea was to kind of look at something which is very relevant in the study of genetics, specifically now in the past few years, and the genomic sequencing of cancer is definitely quite prevalent. But because of that, we encountered quite a few problems, being that there wasn't enough information and simplified information for someone who was kind of new to the study of genetics, and I think that's something that we definitely found difficult. What I would definitely say I would change next time is a more solid plan for our video and a better structure. But what I really liked and what I liked doing was making those little funny, creative bits throughout our video, trying to engage, the target audience of high school students. But overall, it was quite rewarding, and I enjoyed working with my team, and I would definitely do it again.